All right, welcome everyone. My name is Matt. I'm a naturalist at Cleveland Metro Parks. I'm based out of the Watershed Stewardship Center, which is in the West Creek Reservation in Parma. And tonight on School of the Wilds, what we're going to talk about are creaky crawlies. So we're going to switch on over here. There we go. So we're going to discuss creaky crawlies. And as I go through, you're welcome to ask any questions. It's possible that I might miss them as they pop up. And so if you're interested in the kind of almost bottom right-hand corner of my slides, there's a tiny little slide number that you might be able to see. And so you can use that slide number, kind of write that down. And that way, if you have any questions that I was unable to answer during the presentation, you can reference back to that slide number. And that way I know which one you're talking about, so you don't have to just say, you know, the slide with the bugs on it, because it's going to be a lot of the slides. So I might get a little confused if that's your only clue for me. So that being said, I'll be able to go through my presentation. We'll definitely open it up for questions at the end as well. But feel free to write any in the chat now, and I'll try to get to them as we go through. Otherwise, I'm happy to answer them at the end. So let's begin. So first off, what are creaky crawlies? What do I mean by that? So we've got a few pictures here. And the first thing we need to know about these is that they are aquatic. So aquatic meaning they live in the water. And the next thing we're going to break down is the next word is macroinvertebrates. So we've got macro, so not micro, so we use microscopes to see small things. Macro would mean the opposite then. So macro are large things, so they're visible to the naked eye. We don't need a microscope in order to see them. And then finally, we have invertebrates. And so invertebrates are things without a backbone. Right, so we're looking for things that are living in the water that we can see without using a microscope and that do not have a backbone. So we all have backbones, we're vertebrates, unless any of us have left it in our other shirt. You know, feel free to check, make sure you still got yours. Mine's still there, so we're all good there. So we're looking for things without a backbone though. And another good question is, why would we want to study aquatic macroinvertebrates? Why would we want to look for these things? Besides the fact that they're just cool. But there are other good reasons besides that they're just awesome. And it's fun to, you know, kind of flip some rocks and see what you can find. But one of the main reasons is that aquatic macroinvertebrates are a really great bioindicator. And that means they're able to tell us about the environment and the health of the environment. So you might have heard that word before, bioindicator, or you maybe have heard the kind of saying, you know, the canary in the coal mine, right? So that canary was kind of a warning for early miners. If, you know, there was a gas leak, they knew to get out of the mine. So we can use different species to kind of give us an overall quality of the environment. And macroinvertebrates are one of those great species that we can use to do that. And that's because some of these macroinvertebrates are very tolerant to pollution or low oxygen conditions in the water, while others are very sensitive to those conditions, and they really need nice, cool, clear, well-oxygenated water. And so by sampling a stream and seeing what our biodiversity is, the mix of different creatures that we find, we're able to determine, based on their tolerance, the overall health and quality of that stream. So if we only found very pollution tolerant species, we know that that stream's not super healthy. If we found a mix of pollution tolerant with some in the middle and some that are very tolerant or sensitive, then we know that our stream's actually pretty healthy. So as long as we're finding those sensitive species, that means our stream is healthy. So remember, you can still find those pollution tolerant species in a healthy stream, because they can live in a healthy stream. They just have better adaptations to survive in unhealthy streams as well. So it's not the end of the world if you find some more, you know, tolerant species in your stream, as long as you're still finding those sensitive species. In addition, they provide a short and long-term effect of kind of pollution and environmental conditions. So if you did a chemical test of your stream, that's more of a snapshot in time. And so you're only going to really be able to get that exact moment that you sampled that water. So if, you had your dastardly villain coming up, twisting his mustache after you and pouring in some kind of chemical after you've taken your sample, that might not show up. That's not going to show up in your chemical test because you've already taken your water out of the stream. And so you wouldn't know that that's affected your stream. Now you could determine that impacts by testing fish in the water, but fish are a lot more mobile than our macroinvertebrates, and so they're able to avoid those conditions. So they can swim upstream or downstream if there's a major issue going on with the quality of that water. Macroinvertebrates aren't able to as easily swim upstream and downstream. If you're thinking about a tiny little, invertebrate, it's a little harder to really move upstream if there's a good flow or downstream without being washed away. So they kind of stick in the same general area in their stream for their life. And that's what allows them to be such a great indicator, because if there's an issue right in that spot, it's going to impact that diversity of those macroinvertebrates for an extended period. So if you're sampling the macroinvertebrates over time, you'll be able to determine the overall health and quality of that stream. Another great reason to study macroinvertebrates is that they're very easy to sample. So there's a variety of methods for sampling them, and we'll talk about some a little bit later, but you can see in my bottom picture on the right there's some yellow nets in the stream with some folks there catching some macroinvertebrates. But most of these methods are actually really inexpensive. They're pretty easy, and they really require very little training or supplies. And that's why they make another great source for scientists to study to determine the health of streams, because you don't need an expensive test kit or the training to use that. You don't need to really be able to identify like a fish down to species. Even these, you're gonna find out that we don't need to identify them down to species. There's kind of a quick, easy way that we can identify them that gets them identified enough that we're able to determine the health of the stream. And finally, macroinvertebrates are very abundant. We find them in pretty much all of our waterways, not just here in Cleveland Metro Parks or in Ohio or in the US, but around the world. And I've worked with students who are based out of Costa Rica, and they've come up here and they've sampled, and they say that a lot of the macroinvertebrates we find are very similar to the ones that they find. And really, one of the only main differences that they saw was that instead of crayfish, they have crabs in their streams. So I think that'd be kind of cool, but I'm glad we're not losing our crayfish in exchange for crabs, because I think crayfish are pretty awesome as well. So they're great bioindicators. They're really easy to sample, and they're very abundant. So it makes it really easy to really sample these species to determine the health of a stream. So we have two basic types of macroinvertebrates. And the first type we're going to talk about are insects. And so a lot of the macroinvertebrates we find are larvae or nymphs. So those are the young stages of insects. So they're going to mostly be fully aquatic. There are some that are semi-aquatic though. And then there's some that are larvae. So they're going to be caterpillar or kind of maggot-like. And so that's going to be like our center photo on the upper row there. And that is a black fly larva. And so you can see it doesn't really have any little legs sticking out like the others do. It definitely looks a little more kind of like a maggot, right? And so that black fly larva actually has on that larger round end, it's got like a little suction cup that it uses to stick on the rocks. And then the black shiny head there has two little fans that stick out of its head that help collect debris that's floating down, and that's what it eats. So it'll just stick into those little fans, the little organic material, and then it can just slurp them right off. Sometimes I wish I could just eat that way. I can just stick some little fans up and just have a pizza just kind of stuck in there, and it'd be a lot easier than cooking. But I guess pizza doesn't arrive that way, so I'd be stuck just with whatever's floating downstream. So besides our larvae, we also have nymphs. And so we've got two examples of nymphs shown here on either side of that black fly larva. So on the left, we have a stonefly nymph, and on the right, we have a mayfly nymph. And so both of these look pretty similar to the adults. The adults will be terrestrial, though, and have fully developed wings. And so we can see that the wings are not present, or at least fully developed wings are not present on these nymphs. But if you look at the stonefly, the one on the left, you might be able to notice that there are some small, let's go back there. There are some small wing pads right there that you can see. So those wings are developing, and they'll eventually, as that nymph grows, they'll continue to develop, and that nymph will eventually emerge out of the water, shed its skin one final time, and become terrestrial with those fully developed wings. In addition to larvae and nymphs, we also have adults. And so we have some that are fully aquatic. So things like the Riffle beetle larva, so that's the one on the left. So that'll just be tumbling around in the water, crawling over the rocks. And then we have some that are terrestrial, so the dragonfly. So I think a lot of us are pretty familiar with seeing dragonflies fly around. So their nymphs, their early stage of life actually begins in the water. But all insects have 3 body parts. So they're going to have a head. So I'll try to point those down here. We got our head, our thorax, and our abdomen. And so we have those 3 body parts there. We've got 3 pairs of legs, a little easier to see on our beetle here. They'll have a pair of antennae right there as well. And then typically 1 to 2 pairs of wings. So you can see those on our dragonfly. Our riffle beetle does also have wings. So they're hidden under that elytra, that harder outer shell that beetles have. So when we're talking about insects, we have those 2 different types. So we have some that have that larval stage and some that have the nymph stage. And that's based on whether they go through complete metamorphosis or incomplete metamorphosis. So a complete metamorphosis is what we think of when we think about butterflies. So they start as an egg, they'll hatch, they're a larva or that caterpillar stage. After they eat and eat and eat and grow and grow and grow, then they'll form a pupa or a cocoon, depending on their species, right? But they'll form that pupa stage. And that's where they kind of are able to break their body down, rebuild it, and then they can emerge as an adult. Incomplete metamorphosis, on the other hand, they start as eggs, they're going to hatch, and they're going to look pretty much like the adult, but just a very small version. And they're going to eat and grow and eat and grow and shed their skin as they grow, getting larger and larger and larger until finally they'll be able to crawl out of the water, shed their skin one last time, and then their wings will unfurl and they'll be a winged adult. Now, a lot of our macroinvertebrates within this life cycle, they really only live 1 year. Some of them can live multiple years in a nymph stage under the water. So dragonflies are a really great example. Some dragonfly nymphs can live 5 to 7 years before they emerge as an adult. But a lot of these, like our caddisflies and mayflies, really only live for 1 season. So they spend a lot of their time growing as the nymph or larva. And as an adult, they're really only around, sometimes even for a week or so. A lot of them don't have a mouth part as an adult, so they're not feeding, which means they're unable to eat. So our second type that we're going to talk about are non-insects. So we had insects, and now we'll talk about non-insects. So we have crustaceans, so things like our crayfish. They're going to have those 2 pairs of antennae, those kind of pincers, and they'll have that thorax covered by a carapace, so that hard kind of shell. Next, we have mollusks, so everything from your freshwater clams, and there's a bunch of different species of freshwater clam that we can find in a lot of our streams, all the way down to our tiny little fingernail clams, which is what I have pictured there. So that's full size in that little paper clip. We also have flatworms, so planaria. And you can see that little kind of arrow-shaped head with a little white kind of eye spots there, those little proto-eyes that really just help them really determine light and darkness. They can't really see much else with those. And then finally, our annelids. And those are our segmented worms. So things like an earthworm, except earthworms live in the earth. So we have things like aquatic worms, and also everyone's favorite, the leech. And that's what we have pictured here. And a quick side note about the leech. Not all leeches are parasitic, so not all of them are going to attach to organisms and suck blood. There are some that do that, but there are some that aren't. And it's a quick backstory. I discovered one back when I was a scout, and we packed up our tents after a really wet summer camp. And underneath our tent, there were these, I mean, they must have been 2-3 inches long at least, kind of jet black with a little yellow racing stripe down them, these giant leeches. But those are actually non-parasitic, so that was good. I didn't know that at the time. I just saw a giant leech, and it was kind of freaky. But learned since then that those actually eat earthworms and some other insects and things. So nothing to be worried about. They were actually pretty cool, and I've seen a few of them since then. So there's all sorts of things that you can find in our streams and sometimes crawling out of our streams. All right, let's move on. So we're going to talk about our functional feeding groups, because that's another way that we can kind of classify our macroinvertebrates, and it helps us determine the health of our stream. So first we have shredders. And so they're going to break down large organic material. So in a lot of our headwater streams, those are our tiny little streams that are the kind of first little streams that start flowing and then will combine to form our larger creeks and rivers and so on. Leaves falling into those headwater streams from our trees, that's that first building block of our food webs, of our food chains and food webs. So there's not a lot of aquatic macrophytes, plants growing in the water. You can get some algae a little bit as well, but typically they're going to be shaded out, and so you're not getting a lot of sunlight in, so you're not going to get a lot of algae growth either. So those leaves falling in are really important, but a whole big giant leaf is too much for a tiny little macroinvertebrate to eat. So there's shredders who start kind of chomping away, and they'll break off pieces of that. And as they break off pieces of that, they're going to be able to consume some, but some of those pieces go floating downstream. And so then we have collectors. And so we have collectors that, like the black fly larva I was talking about earlier, they have those little fans on their head that collect things that are floating down. So they're going to collect that organic material. So you can have coarse particulate organic material, or CPOMs, those are larger pieces, or you can have fine particulate organic material, or FPOM. And so those pieces float down, and you'll have different organisms that are able to collect those and gather them up and eat them. And so the example in our picture here is actually a little net spun by a net spinning caddisfly. And so the little pieces of that organic material floats down, gets stuck in that net, and then the caddisfly is able to pick it off, just like a spider would pick off prey from its web. We also have scrapers, excuse me, and so they're going to scrape along rocks and logs that are in our stream. They're going to feed on algae and diatoms and other biofilms that are growing on the surface. If you've ever held a larger snail before, you might even feel it kind of scraping your dead skin cells away. In some places you can even get little, manicures, pedicures where the snail's just kind of eating away at your dead skin cells. So I've never had that experience, so I don't know. If you've had that, you can let me know in the chat later. Let me know what you think about that. And then finally, we also have predators. So these are organisms that are going to hunt our other macroinvertebrates, and there are even a few that will hunt larger prey. So anything from smaller fish, tadpoles, even adult frogs. These macroinvertebrates live in a bunch of different habitats within our streams. And so I just wanted to touch on those really quickly in case we're unfamiliar with some of those different habitats. So a healthy stream should have all of these habitats. So they should have your runs, riffles, and pools. A lot of times in our urban streams, so for example, here at West Creek Reservation in West Creek, because it's such a heavily urbanized stream, and our watershed, the area surrounding our stream, has so much impervious surface, so surfaces that water can't soak through. We don't have enough groundwater feeding our stream to keep it at a deep and fast-moving stream. And so what happens is we get a very just shallow, slow-moving stream, and that habitat's called a glide. And it's not the best habitat to really provide for the best biodiversity of life, whether it's macroinvertebrates or fish populations. And so you'll see that in a lot of our more urbanized streams, that it's more just kind of a slow, shallow moving stream instead of a kind of faster moving stream that has these different habitats in it. So we're going to dive into these habitats here real quick. So runs and riffles. So in the kind of background here, we have more of a run. And so that is a deep, fast moving area of water. And you'll get different species that exist in these different habitats. And then in our riffle, which is in the foreground here, you'll find a lot of macroinvertebrates there, because that's a very shallow, fast-moving area. And as the water goes over those rocks, it's pulling air in and it's adding oxygen to the air. So this is a really well-oxygenated part of the stream. It is a faster moving area though, so you do have to have macroinvertebrates that are well adapted to clinging onto those rocks. But otherwise, you get a lot of oxygen coming in that area, just like in our fish tanks. Why our filters will pour in, it's adding oxygen that way, or sometimes we have bubblers in our fish tanks. So this is nature's bubbler. Next, we have pools, and so those deep, slow-moving areas. You can find different macroinvertebrates living in here. A lot of times you'll find deeper or larger fish living in these deep areas. And so it's another different habitat where you can find different species that exist in there. And then finally, we also have marginal areas, and it depends on your streams, and how intact that floodplain is. But here you can see we've got a lot of aquatic plants growing right along the sides, right in the shallows. And that's going to allow a lot of macroinvertebrates to hide in and among those plants. So especially things like damselflies really like those kind of habitats as well, where they can kind of cling onto those plants and blend in and camouflage to help protect them from fish that might want to eat them. And so it's a great habitat for them to survive. So we have our runs, riffles, pools, and then our marginal areas as well. So again, why do we want to study macroinvertebrates? So again, they are a great bioindicator. They're going to tell us the health of a stream. And that's important because it's an easy way to determine the health of our stream. Water is important to everyone. And in fact, part of the reason why I focused this presentation tonight on aquatic macroinvertebrates and stream health is because next week on March 22nd is World Water Day. And that's a whole day about celebrating water around the world. And we usually have a big celebration here at the Watershed Stewardship Center. Unfortunately, because the centers are still closed, we're not having our traditional celebration here in the center. But it's still a good thing for everyone to check out. If you go to worldwaterday.org, you can see what's happening around the world, and you can participate as well. They're doing a lot of online activities this year. So I highly encourage you to check out worldwaterday.org to see how the rest of the world celebrates water as well. So aquatic macroinvertebrates, again, they're a great bioindicator. They're easy to sample, and they're very abundant, so we can find them everywhere. And let's take a look at some of that pollution tolerance. So on the left-hand side, we have 3 groupings of macroinvertebrates. And in our first group here, those are our sensitive species. So they really need the cool, clear, well-oxygenated water. And in particular, the 3 we want to focus on are Mayflies, or Ephemeroptera, our Stoneflies, or Placoptera, is the Latin name, and then Caddisflies, Trichoptera. And so a lot of times when you're talking to macroinvertebrate scientists, they'll abbreviate those as EPTs. So Ephemeroptera, Placoptera, and Trichoptera. So your EPTs. So you want to find those EPTs for a nice healthy stream. In the middle, we have our second group, and these are kind of mid-range species. So they can survive in waters with slightly lower oxygen, maybe some temperature differences. Maybe we get some more warm water flushing in occasionally, but nothing too severe. So things like our crayfish, so we find those in a lot of our streams, right? Aquatic sow bugs, so this guy, so the little potato bugs, roly-polies, pill bugs, whatever you call them. So those are crustaceans, and there's an aquatic version that we have in our streams as well. If any of you out there like shrimp, the scud here is a little freshwater shrimp. However, they only get to maybe like a centimeter large. So if you were thinking you're gonna, go down to your stream and collect a bunch for a good hearty meal, you'd be out there for a little while, I think, collecting some scud to eat. So I wouldn't recommend it. Now you may have noticed if you're looking at some of these other organisms in group 2 that we have another caddisfly here. And I said earlier that our caddisflies are in group 1, which we do have some, but we have some caddisflies, especially our net spinning caddisflies, these filtering caddisflies, the Hydrocyclides genus, that are a lot more tolerant than most of our other caddisflies. So you will find these caddisflies in our streams a lot. They're typically kind of greenish brown color. And again, they're those net spinning caddisflies. So kind of like we talked about earlier, how they'll spin that little net to catch that organic material that's floating downstream to consume that. So we do have some caddisflies that are very sensitive, some that are a little more mid-range. And then finally in group 3, this is where you have your midge larvae. You can get aquatic worms, pouch snails. and a few other kind of fly larvae and things. Typically a lot of things that can survive very low oxygen conditions, whether they're well adapted to breathing those low oxygen conditions, or they have extra adaptation that allows them to actually breathe the air, so they don't have to worry about low oxygen in the water. So we take those pollution tolerances, and now we have to figure out, okay, well, what does that mean? How does that tell us the health of a stream? And so the Ohio Department of Natural Resources has a stream quality monitoring program. So their Scenic Rivers program, so you can look that up later if you're interested in trying to take part. And I'll talk about a few other opportunities later on as well. And you can see we have, again, those three groups. And so what you're doing is when you go out and sample with them, if whatever, macroinvertebrates you find, you would place a little letter grade. So if you found 1 to 9, it's A, 10 to 9, B, 100 plus is C. So let's say, we found, I'm just going to do a little check mark. We found mayflies, stoneflies, and caddisflies, right? So those EPTs. So we found 3 of those. And so we've got 3 here. We multiply that by 3, and we get 9. Let's say with our middle group that we found crayfish and scud and sow-bugs. So we take those 3, multiply those by 2, we get 6. And at the bottom, let's get some lovely leeches in there, and let's get some aquatic worms and black fly larvae. Why not? So we got another 3, but this time we're only multiplying them by 1. So that's just 3. We add up our total score there. So if you're better at math than me, maybe you've already done that, but that's 18, unless I'm wrong, but I'm fairly certain that's right. Did this on the fly. So that falls into this 17 to 22 range. So if we sampled a stream and that's what we found, then our stream quality comes out as pretty good. That's not too bad. So that would be a good rating we'd like to see. Obviously, you could get a little higher, and that's nice too. But you might not find, all the species that exist in that stream every time you sample. And that's why with this program, they like you to sample at least 3 times a year. So usually kind of late spring, middle summer, and sometime in the fall. They don't really encourage you going out in the winter because it gets really cold in the water. So we don't want to do that. All right, so that's how we use our pollution tolerance to determine the quality of our water in our streams. So again, I was talking about some of these adaptations that these organisms have in order to survive these kind of low oxygen conditions and other conditions, and just being able to survive in streams in general. So I wanted to showcase a few of those. So the first we have here, so this is a rat-tailed maggot. So from the name maggot, you might be able to piece together that it is a fly larva. And you might be able to see that, it's got this nice kind of long skinny tail, and that's why it's named the rat-tailed maggot. But that tail actually is its breathing apparatus. And so it's a little snorkel. And so it will stick that little tip out up above the top of the water, and then it's able to breathe the air, and that way it doesn't have to breathe the low oxygenated air or low oxygenated water, and so it can survive in those low oxygen waters. And I did see a question come in, and it was about what does the state do with that data, and that's a great question. And I should have mentioned that we're going to get to that near the end of our presentation. So so hang on to that question. We'll get there. All right, next we've got one of our lovely beetles here, and you can see it's got these really lovely large flat paddle-like hind legs, and that's really going to allow it to just shoot through the water really quickly. So a lot of these beetles are predaceous, and so they're going to be chasing after other macroinvertebrates to consume, and so they need to be able to move really quickly and make sharp turns. And so by having those really large paddle-like legs are really going to help that move a lot faster through the water. All right, this is another one I've talked about a few times already, and we've got our net spinning caddisfly, but this is a nice close-up look, so you can really see that little spun net. And this right here, this is the head of the caddisfly, so the rest of the body is further down in this little kind of case that it's made, but you can clearly see that net there, and that's what's going to catch and hold on to that organic material as it's floating downstream. And then the caddisfly can just safely stay inside of that little area and just kind of chow down on the food as it comes to it. We also have case building caddisflies. And so as you can see here, this one's used its silk instead of spinning a little net, it's able to create a little case made out of small rocks that it's attached together. And it uses that to protect itself as it travels around looking for other food in a stream, or sometimes in wetland systems, pond systems as well. What's really neat about some of these is there are people who actually will farm different species of these case-building caddisflies, because these caddisflies will really use, some of the species will use whatever they can find to make their case. And so instead of putting in little rocks, they'll put in flecks of gold and little gems and things, and the caddisfly will actually make their case out of that material. And then you can carefully remove the caddisfly from the case, and then they're able to make that case, that former case, into a necklace pendant or earrings or other jewelry. And so I thought that was kind of neat that you can, kind of, keep these little guys and keep them happy. And I mean, eventually they're going to, mature and evolve, and so you're going to have adults flying around. So you're going to have something over top of the container so they don't fly away. But otherwise, you can get these lovely little cases made here. So let's talk about how we monitor them now. So we know they live in the stream. We know their tolerances. We know how, that breaks down. We can use them to determine the quality of our stream. So how do we actually catch them? So one of the most basic ways is using a kick net. You really need at least 2 people for this process. So you can see we've got someone holding our net there. They've got it so the water's going to flow into our net. And you can see it's at an angle as well. So you don't want it going perpendicular to our stream, because then it's going to act more like a trampoline for the bugs, and they're just going to bounce right off. So having it at an angle allows that water to flow through the net, and any macroinvertebrates are going to get stuck on the net instead of bouncing off. And you'll have someone standing in front of that net, and they're going to do the stream shuffle. And so they're really just going to kick their feet around and really just kind of spin around as much as they can and kind of kick up all that sediment and rocks, because that's where those macroinvertebrates are living. They're underneath those rocks and sediment, because if you're a tiny little invertebrate, there's fish, frogs, bigger invertebrates that are coming to eat you. So you really want to try to hide. So they're typically in that sediment and under those rocks. So you really want to kick all those up in front of that net, and the flow of the water flows into the net, carrying those macroinvertebrates, and then you're able to lift your net out, take it ashore, and see what you've caught. If you want a little more versatility in what you're catching, or you're just by yourself, and so kick net would be more challenging, you can use a D net. And so you can see it looks just like a D. You would put this flat part on the bottom of the stream, so your water's flowing into it. And again, you'll do the stream shuffle and kick around in front of it and let all that water flow in, carrying the macroinvertebrates as well. The D-net's nice as well because you can kind of poke it into overhanging banks, somewhere where you might not be able to really do your stream shuffle, or into deeper pools, and kind of scrape along the bottom. And that way you can catch a wider variety as well. If you need more of a quantitative sampling instead of qualitative, the server sampler here has a little metal square frame. It might be a little hard, so I'll just kind of draw our square there. Has a little square bottom frame there, and so you're only sampling what's in that square frame, and again, it's going to flow into your little net. So that allows you to make sure you're sampling the same size every time that you sample, so you can get a more quantitative sampling instead of just a qualitative. Depending on the type of study you're doing, it could matter, but for basic stream quality, you can just use a kick net or D net, and you're able to just use that qualitative sampling. So another method is the leaf pack. And you can take a bunch of dried leaves and throw them into an old onion bag or clementine bag there, like that, and stake it down into a stream. The staking it down part's important, especially if you live in a very flashy stream system. So flashy meaning a lot of stormwater rushes in and then rushes out. So you want to make sure it's staked in there well, and that you remember where you staked it. And so you stake your leaf pack in there, and you leave it there for 6 weeks. And what's going to happen is you're going to have macroinvertebrates move into that leaf pack so they can start consuming the leaves. And as you have those shredders moving in to consume the leaves, you'll have your collectors moving in to collect the little pieces that are coming off. You'll have your grazers, your scrapers moving in to eat the biofilms that are developing on there. And you'll have predators moving in to eat all the other macroinvertebrates moved in. So you can get your whole little ecosystem kind of self-contained in that bag, and you can collect it later, bring it back to your lab, classroom, whatever, and open it up and then sort through it and find what you have. Kind of similar, a little more scientific method of it is this Hester Dendy. So it's these little discs kind of on a screw, and macroinvertebrates will kind of move into those small spaces to hide and survive. Plus biofilms will develop on there, so you'll get a lot of scrapers moving in, and then your predators will move in to eat those as well. So that whole cycle again. So we've collected our macroinvertebrates, and now we have to identify them. And honestly, to get to the level where you just need to determine the health of a stream, this is all you need to know. The basic head, thorax, abdomen, be able to identify antennas, eyes, foreleg, middle leg, hind leg, wing pads, where the gills are, tails. I mean, that kind of terminology is really as far as you need to go for our basic, stream samples. I have more background in this, and so I had to use, the big kind of textbook version of it. And so I'm going to read one of the options here. So it's basically presented as a dichotomous key. They're given two choices, and so you have to study your insect to determine which of the two choices it most resembles, and then you follow it down until you get your answer. So here is one of the questions. So we're identifying a type of mayfly. We're trying to get it down to genus, and it says body dorsoventrally flattened. Posterolateral projections of abdominal terga 7 and 8 longer than the median length of respective terga, 4 femora wide, width 3/4 the sub equal the length, 4 femora margin with long setae, dorsal surface of 4 femora with row setae across proximal third, inner lobe of ventral lamella of gill 2 present or absent, when present inner lobe length variable up to 1/2 of outer lobe. And then the other option is just not that, basically. So you can see it gets a lot more complicated if you're going a lot further in. And so unless you kind of really geek out about it like I do, you probably don't need to worry about that. That's a little much. So the easier version is right here. And this is what we use with a lot of our students. And again, this is going to get you to that level that you need in order to determine stream quality. And so we have our dichotomous key in the center, and we've got our organism on the right. And I don't know what that organism is. That's a cool looking thing, but I have no idea what it is. So let's see if we can figure it out. So we're going to start at the top, and we're given two choices. So we're going to have to determine if our organism has a shell or no shell. And looking at this, it clearly does have a harder, carapace, that outer, that exoskeleton, but it's not a shell. It's not like a snail or clam. So we're going to go with no shell. So then next, we have to determine if it has legs or no legs. I think we can all clearly see it has legs, so let's follow that path down. All right, and so now we're given three choices here. Does it have 10 or more legs? Does it have 4 pairs of legs? Or does it have 3 pairs of legs? And so we can easily count those up. We've got 1, 2, 3, there's a little one hiding under there, 4 and 5 on the other side. So we've got 10 pairs of legs, so 10 or more legs, not 10 pairs, 5 pairs, 10 or more legs. And then finally, we just have to see if it looks most like species 1, species 2, or species 3. And I think we can all tell that if we look at our little clues here, lobster-like, well, that definitely looks kind of lobster-like to me. And so, yes, we have a crayfish. And again, this nice, simple, easy dichotomous key is what we use with students. It gets you what you need to know in order to determine the health of a stream. All right. So I wanted to talk very quickly about some of our species. This was the hardest part for me to make this presentation because there are so many cool species and so many great pictures you can find on some websites, which I'll give you the site names later at the end of the presentation, so you can go and check it out because they're just so great. But mayflies. So I wanted to focus on our EPTs, the mayflies, stoneflies, and caddisflies. So the diagnostic characteristics are they're going to have abdominal gills, so they're going to have gills on their third body segment, a single tarsal claw, so that just means at the end of their legs they'll have one little claw, and they usually have 3 tails. And a very common mayfly that we find in a lot of streams are the flat-headed mayflies, the Heptageniidae. And so you can see that head is very, very flattened. The eye is really kind of on top of the head. And that's because these really enjoy living in riffles. And so there's a lot of fast moving water. And so they need to be able to stay very flat on those rocks and cling to those rocks as that water just rushes over them. Something I wanted to point out too is you can see the gills here and these little gill plates covering the gills. So the gills are the kind of little kind of fluffier parts underneath, and then they've got some of those gill plates there helping protect them. So those are those gills on the abdomen. And again, we've got our 3 tails, 1, 2, and 3 on our mayfly here. Those 3 tails are probably one of the most easy identifiers for them. That's what we typically teach students, that, you know, it's got those 3 tails, it's forming an M, and M is for mayfly. And so that's a nice easy way to keep that in mind. For other species of mayfly, we've got our batids, and these are minnow mayflies. So these also like fast moving water. Again, you can see the 3 tails, you can see the gills on the abdomen, and these mayflies have a very fusiform body. And so it has that kind of S shape, and that really helps them swim through faster currents. And so that's why these are the minnow mayflies, because they kind of swim like little minnows as they're moving through the water. All right. The prong-gilled mayflies, or Leptophlebidae, these guys I've just always enjoyed because their gills do look so much different than others. So, you know, we've got one single gill right there, those little prong gills. I just think they're kind of cool. But again, I geek out about this stuff, so that's all right. These are another one of my favorites, the Ephemeridae, or the burrowing mayflies. These guys are extra awesome because you'll notice they actually have little tusks up front on their head there, and that's what they use to help kind of burrow into typically sandier sediments in order to hide from prey. You'll notice too that their gills are a lot you know, bristlier, fluffier, and so they've got more surface area to help make it easier for oxygen exchange. Because if you're buried down under sediment, you really need to be able to access that oxygen at a higher rate than if you were just out in the flowing water. I'm going to move through these kind of quick just because I want to make sure we end on time as well. And I could you know, obsess over these all night. Stoneflies, or Placoptera. So their diagnostic characteristics are they'll have 2 dorsal claws, so 2 claws at the end. And you'll remember that our mayflies had 1, and they'll usually have 2 tails. It's tricky sometimes when you're sampling because sometimes those tails are fragile and can break off. Same with the mayflies. So that can be tricky if you're working with students and they're like, well, this only has 2 tails, so it's not a mayfly. But usually you can see that one's broken off. But here's an example of a stonefly. So the perlids, these are our common stoneflies. Although they are called common stoneflies, they do still really prefer nice, cool, clear, well oxygenated streams. So they need to be in more of our kind of excellent quality streams, typically. They're one of my favorites. I really love the patterning on the back of the thorax. I just think they're cool little critters. Their gills, it's kind of hard to see with this white background. They have kind of little fluffy gills kind of in their armpits. And when you collect them, if you have them in a tray, or well, and in nature, when they have low oxygen conditions, they do little push-ups, and that helps cycle water underneath them and through their gills. So they do little push-ups, and so you can notice that. Typically, though, you know, if I've out sampling and I notice that, I try to get them released quicker or into some new water because I don't want them to be stressed out. Another stonefly are our isoperlas, or our striped tail stoneflies. So you can definitely see that kind of striping on the abdomen back there. But you'll also notice the wing pads. So those wings are developing. So this again is a nymph, and will eventually emerge as a full adult. You might also notice these kind of 3 dots on the head there. And so again, those little oculi help kind of sense light and dark, so they can help kind of determine if they're hidden or not. So another just little feature that you can notice on some of these macros. Caddisflies, or Trichoptera. So their distinguishing characteristic are prolegs with a single hook on the end. So at the end of their abdomen, they'll have 2 little prolegs, which will be kind of like little nubby caterpillar legs. And they'll have a little single hook on each of those. So our Hydrocyclides, those are our net spinning caddisflies again. So I wanted to bring those back. Again, when we typically see those, they're usually a little further down. So they've got these first few segments here are sclerotized, which means they have that harder outer shell. But further on the abdomen, they're non-sclerotized, so it's just that soft body. And typically they're a very like bright green color, sometimes brown, depending on the species and their diet and things. Moving on to case-building caddisflies, we have our Limnophilides, or our northern case makers. I have a few pictures of these because our Limnophilides will use different materials to build their cases. So this one you can see is used really kind of flat pieces in order to build its case. Also, I think just the colors on this caddisfly are just gorgeous as well. They really coordinated their outfit, and I just give them props for that. I think that's really awesome. But another Limnophilid we have. So this one here, you can see is used little shredded pieces of plant material. And so there, a lot of times we find these in our wetlands here at West Creek and the Stewardship Center. And then another one, like I said, they'll use whatever materials they can find. So this one's clearly in an area with a lot of different little snails, and it was able to use those discarded snail shells to make its case. So, you know, whatever they can find. All right, so now we're going back into what we do with that data, right? And so that was a question someone asked earlier was, if you're participating in that state's scenic rivers program and collecting data for them, what do they do with that data? So some of that data in Ohio is incorporated into this. This is also from US EPA, where they've sent scientists out. So this is from 08-09 data. And they're compiling that data together to determine the overall health and quality of streams in different regions. And so the kind of green shaded regions all have a similar kind of habitat. And so those are our eastern streams. The kind of beige are the plains, and the orange are the west. And so you'll notice in the east, about 63% of our streams in 08-09 were rated as poor. And again, you got to keep in mind that it's a very heavily populated and urban area. And so we have unfortunately impacted our streams pretty negatively due to that. Although we still have 17% that are rated as good, so that's good. But then in the plains, they've got a slightly smaller percent rated as poor, only about 58.5, but also a slightly smaller percent rated as good, 16. Out west, only 30% rated as poor, and 42 basically percent rated as good. But remember, a lot of that land is undeveloped. A lot of it is federal property, and so there's not a lot of development going on out there. So their streams are a little more intact and healthy. So it is kind of hard to just compare region to region, but it's good to know where we're at. And overall in the US in 08-09, we had about 55% rated as poor. And so definitely not something we really like to see. It'd be nice if our streams were healthier, and we'll talk about how we can kind of help with that in a moment as well. There is a little more recent data. This is from 2013, 2014. Unfortunately, I couldn't find a nice little graphic like they had in 08-09, and so I'm providing you with this very scientific chart here. But you can see on the left-hand side of the chart, it's showing you the percentage that were rated good, fair, and poor. And this is overall across the country, not broken down by region. They did have it broken down by region, so you can go to the US EPA's website and look at all this data if you'd like. They do have it broken down by region, but they added many more regions, so I think it was like 7 or 9 different regions, and I didn't want to have to dig through all those for us, so I decided let's just look at the overall US data. So again, we've got about kind of 30-ish% rated as good, about 25 to 30% rated as fair, and about 45 or so rated as poor. The good news is you've kind of look at the change. It looks like, I've got slightly more rated good, slightly rated fair, and slightly less rated poor. Unfortunately, none of those are statistically significant, so there wasn't a scientifically significant change in those ratings. So that's not necessarily all bad news, right? While it would be nice to see things improving and having more good quality streams, at least we're also not slipping backwards. So the fact that we're still able to kind of keep the same percentage rated as poor can be seen as good news. So what can we do then, right? If we don't want to have these poor rated streams, what can we do? And so one of the things you can do is protect your watershed. And so you can install stormwater management features on your house. And so those are things like rain chains, which are attached to your gutter instead of downspouts, letting that water trickle down and then soak into the ground. Rain barrels, so you're collecting water off your roof during really wet seasons like spring, and then you're able to use that water to water your lawn or garden. You can use it to wash your car if you wanted. But basically, we're collecting that water, slowing it down, and letting it soak into the ground. Instead of just going into our downspouts and into our storm sewers, where it's going to end up rushing right into our streams, causing erosion, carrying runoff, making those streams really flashy, making it really hard for our macroinvertebrates to survive, as well as other organisms. You can also try to encourage your place of employment to install stormwater management features. So, if you're good at home and, you've already got all your rain gardens and your rain chains and your, porous pavement and everything, start reaching out further into your community and trying to encourage other places to do this. And it doesn't have to be your place of employment. It could be your actual community. Talk to your city council and get them to agree that we should allow rain chains and rain barrels, because some communities are still a little behind on that. So you can work, with your neighbors, your friends, your family, your community, try to get more people to adopt these measures to help stop all this water rushing directly into our streams. Other things you can do is join local watershed groups. So I think pretty much every local stream in the Cleveland area has a local watershed group. If you're watching from outside the Cleveland area, there are still a lot of local organizations around. Best thing to do is just kind of do an online search for your area, for your local stream, and just type in your stream's name and put watershed group, and usually that should pop things up. You can assist in collection of data about local watersheds. So a lot of watershed groups will do that as part of their programming. But there's, like I said, the Ohio Department of Natural Resources also has programs. I'm sure other states do as well. I'm not very familiar with them, but I'm sure they have other programs that could be similar. And if you are within Cleveland Metro Parks area, we have our Cleveland Metro Parks Watershed Volunteer program, which really helps with a lot of these issues and helps educate you further as well. And that leads me to my next point, is continue to learn. So if you explore your local waterways and learn more about them, and that can just even be just going out on a hike and just taking the time to really look at streams, and maybe go down to a stream and carefully turn over a rock and see what's living underneath it. Put the rock back afterwards, because you don't want to destroy their home. But you can take a look at what's under there. And continue coming to Cleveland Metro Parks programming. Go to other programming if you're not in this region, or continue watching us virtually. And you can use iNaturalist as well if you're unfamiliar with iNaturalist. It's a free app that you can get on your phone, or you can use it on a computer, and you can take pictures of plants and animals that you see, and it does a pretty good job at identifying them. It might not work so great for macroinvertebrates yet. It's still a little iffy there, but for a lot of other organisms that exist within your whole watershed. So that way you can learn more about what's in your watershed, and you can become more connected with it and be able to speak more about it when you are speaking to people. So protecting your watershed, joining those local groups if you can, and continuing to learn is always a great thing that you can do to help improve the quality of our streams and our environments. And that brings me to the end of my program. So I thank you for joining me. I will open up for questions in the chats. I know there were a few that popped in there, so I'm going to open my chat box back up and answer those. Just real quick, I wanted to give a few thanks. So Claire Weldon is our aquatic research coordinator, and she had put together a PowerPoint ages ago that I was able to look at and kind of build off of, so I really appreciate that. The Stroud Water Research Center, located in Southeast Pennsylvania, has a website called macroinvertebrates.org, and it allows you to get a nice up-close look of these macroinvertebrates, and it's going to be able to show you all the identifying characteristics, going down to genus, so you can really get, dive in deep with that if you want. It's a really great website. I encourage you to look at that one. And then a lot of the nice macro photography in my presentation came from Jan Hamrsky, and I apologize if I butchered that name, but his website, lifeinfreshwater.net. He granted me permission to use his pictures in this presentation, and they're gorgeous. They're absolutely gorgeous. I highly recommend checking out his website to see more just amazing pictures of all of our amazing macroinvertebrates. So all that being said, I'm going to open up our chat box again here, make sure I can answer any questions. So we talked about, so if you do have any other questions, feel free to put those in the chat box. I'm going to open up our question and answers too, in case there's any in there. All right, so we talked about, yeah, so there's a question about what does the state do with the stream health assessment data? Well, I guess I didn't answer fully. So they collect that data, and then they'll use that data to determine which areas they need to focus on to help improve. And so that's the whole point, right? Is, we don't want to just know, okay, well, our stream isn't doing well, the end. Now it's like, okay, well, what do we need to do to help fix this stream? And so that might mean stream restoration in some cases. So that might mean taking a channelized stream, putting meanders back into it, putting those riffles, runs, pools back into it, planting a forested floodplain to help revive that area, make it a more natural flow and a healthier system. And that's all good, but if you're not thinking about the entire watershed, it might be a waste of money, because if your watershed is still really heavily urbanized, you can put all that money into fixing that stream, but ultimately you still have that same issue of all that stormwater rushing in, and it's ultimately going to end up just kind of wrecking it again. Let's see. Yeah, it would be cool to keep the Caddisflies and try to get your own little cases, but yeah, they got to fly off at some point, so it would be a little bit of a challenge, I'm sure. And then students, yeah, so we do typically do macroinvertebrate collecting with groups here at the Watershed Stewardship Center. Typically when we do ours, we will do some stream sampling, but because West Creek is so heavily urbanized, and we also have a unique instance where here at West Creek, part of our land was a former landfill, so they actually rerouted the stream in the 70s around that landfill. And by doing that, they kind of created a pinch point where there's really no floodplains. So because we're such an urbanized watershed, when all that stormwater comes through, it really acts like a funnel. And so it creates a lot of erosion downstream. And so it's really scraped the bottom of our stream, removing a lot of the substrate, the rocks, the sands, the things that those macroinvertebrates need to hide in. And we just have a shale bottom stream. And so it's harder for macroinvertebrates to survive. So we don't typically do a lot of stream sampling with macroinvertebrates here directly at West Creek, unless we have more time where we can take them further upstream. But we do also do stream sampling in our macroinvertebrate sampling in our wetlands, where we're able to still talk about the adaptations they have and trophic levels, so those food chains and how that energy passes as well. Sorry, let me check our question answer box as well. I can see that. Sorry, I had to expand it. Okay. Oh, sorry. Yeah, there's a lot of stuff that popped up in there too. So sorry that you can't see anyone else's questions and things. Yeah, it's set up that way, so they all just go to the host. So yes, so the nymphs do have wings. There was a question, do the nymphs already have wings? So the nymphs do have wings, so they develop their wings slowly in wing pads. So they're not fully developed functional wings until they emerge as an adult. What are invertebrates' different defense mechanisms against fish or other predators? Yeah, that's a great question. So a lot of our macroinvertebrates, they're really good at hiding in those sediments, and they camouflage really well, and they'll hide under rocks and live under rocks. So that's going to help them hide from predators. The case building caddisflies will build those cases. Things like damselflies actually are pretty intelligent, and they can recognize different predators. So if they're clinging onto a plant stem, if there's a fish swimming by, they know they can just rotate around that plant stem to avoid the fish. But that same strategy wouldn't work if there were a dragonfly nymph crawling along the bottom of a stream or wetland, because that dragonfly would still be able to see them. So if there's a dragonfly nymph approaching, they know to release the stem and swim away or get washed away in the current. So they've adapted different strategies to dealing with different predators if just basic camouflage doesn't work. We have a comment here. Net spinning caddisfly looks like it has a mask on its head. Yeah, I mean, again, they're so awesome. Just they can look very different, and they're just very cool. I definitely recommend checking those out. Different stormwater management features, how they work. So I mentioned some of those, but the typical ones you'll see at least in households are rain barrels. So you connect that up to your downspout. You got to make sure that your local jurisdiction allows it. There's different rules and regulations, so check on that first. But you connect it to your downspout, and it's going to collect the water, the rainwater coming off your roof, and then you're able to use that to water your gardens, water your lawn, wash your car, things like that. Rain gardens are also very popular, and you can find some easy instructions of how to build a correct rain garden, because you wanted to make sure it's the right size to handle the amount of runoff coming off of your house's roof. And then it'll tell you the different types of plants that you can plant in there, and native species that are really great at soaking up a lot of water. And a lot of times you talk to people and you say native species and they're like, oh, I don't, those are weeds. And it's like, no, you can get these beautiful flowers, and you can get all different colors, and you can make it so it blooms throughout the whole year, and so it can be constantly shifting, and you can get these beautiful gardens that help collect your stormwater as well. Just became a volunteer. Excellent, awesome. What do the Watershed Program volunteers do, and are they accepting volunteers at this time? So the Watershed Volunteer Program has a whole slew of different opportunities available. So they'll do everything from going out into the field and like collecting data. They'll do lots of different workshops where you learn about chemical water testing, macroinvertebrate water testing. They'll work on you just get a lot of education and training and kind of real life experience. And I believe they're still accepting volunteers. Everything obviously has been a little slow down, a little change right now. I think you can still sign up to be a volunteer for them. I'm not sure the full extent of the activities at the moment, but if you go to clevelandmetroparks.com and look for Watershed Volunteer Program, you'll be able to see what opportunities are available at the time. All right, and I believe that is all of our questions. And so if no one has any further questions, I just want to thank you for joining us. If you want to stick around for another quick moment or two, I have just a few more species I could show you if you want to see some up close pictures. But this does bring us to the end of our presentation, and I really thank you for joining us. And I hope you learned something, hope you had fun. I hope that you are much more excited to flip over a rock in a stream now and really see what type of critters are moving underneath that. I hope that you're more motivated to try to find different ways to become more involved in your watershed, and just keep exploring. It's really great to see what's out there in our world, and really just enjoy being out there. So thank you for joining us. There'll be another School of the Wilds presentation next Saturday, so hope to see you then as well. And I'll end it at that, but if you want to stick around, I'll show you our final few slides. But thank you for joining. All right, we'll move on to these final slides here. Let's see, there we go. Got to close my chat boxes again real quick so I can see our pictures. So we have dragonflies, the Odonata, and their suborder Anisoptera, and this is an Aishnidae, so it's a darner dragonfly. Really cool thing about dragonflies are this mouth part right here, this lower jaw. They can actually extend to capture their prey. So a lot of dragonflies are ambush predators, so they'll slowly sneak up on their prey, and then they're able to extend that jaw. And we actually have a picture of that so you can see. So there's that jaw being extended to help grab their prey, and then they can pull it in to consume it. They'll eat anything from smaller macroinvertebrates, other dragonfly nymphs, to even small fish or small toads or frogs. So they are a very voracious predator, but they're a great thing to have in the ecosystem because after they mature and become adults, they're nicknamed the Mosquito Hawk because they eat lots of mosquitoes. So they're great to have around. Also, just one of the coolest organisms you can see. They're really intelligent. When they're adults, they can track where their prey is flying to and actually intercept it instead of just chasing it. So really, really amazing animals. Here's a picture of our dragonfly nymph with the little fish that it's caught. So you can really see how that arm folds up underneath, and it's just able to really grab its prey to eat it there. And then, oops, this is actually a damselfly, so we'll just ignore that there. So these are damselflies, and this is our broadwing damselfly, or ebony jewel wings. So if you've ever been walking along a stream, usually kind of later summer, and you see a really iridescent green, bluish green damselfly with deep black wings, those are our ebony jewel wings. And so they like to lay their eggs in the kind of faster flowing streams versus wetlands. And so we've got our little nymph here, and these little tails, these are actually the gills of a damselfly. So they're attached at the end of the abdomen there, and so that's how they breathe. But you can find these damselflies typically in those marginal areas I was talking about, kind of clinging to those plants. And then we also have our Lestidaes, our spread-wing damselflies. And I kind of like this picture because I think he's kind of got a goofy little face, but you can still kind of see that jaw underneath and those sharper points of that lower jaw there. So he's kind of menacing in a way, but also kind of adorable. And we see a few other pictures here. So we've got a small damselfly nymph perched on top of this plant. And in the background, we've got that damselfly lurking. And sure enough, that damselfly is able to sneak up and grab that small dragonfly nymph. And that's the circle of life. But I do enjoy that picture because he's really just chowing down like it's a little burrito, just kind of going to town on it there. So, but that really brings me to the end of our presentation. So I really do appreciate all of you joining us and sticking around. And I hope you had fun. And again, join us next week, and we'll see you then. Take care.